Good morning, everyone. Today topic is ribosome and its structures and its types. Generally, ribosomes are granular structures. Granular structure means spherical structures like this, granules. And ribosomes was discovered by George Palade in 1953. And these ribosomes is the smallest cell organelle. Generally, these ribosomes found in both prokaryotic organism and eukaryotic organism. Prokaryotic and eukaryotic organism. Generally, ribosomes are non-bound cell organelle. Non-bound cell organelle means this ribosome they do not covered with any membrane structures. Or simply we can use another term, the membranous cell organelles. Not only ribosome, like centriole, also amembranous cell organelle. Centriole also called centrosome. And not only that, one more important cell organelle, that is this nucleolus also a membrane bound cell organelles. Generally, the centrioles absent in plant cells. In plant cell, the centriole is absent. So, amembranous bound cell organelles are absent in eukaryotic organism, especially in ribosomes, centrioles, and nucleolus. There is an important note. In prokaryotic organism, amembranous cell organelles also there. Here I am writing. Note, in prokaryotic organism, amembranous cell organelles. Here, amembranous cell organelles prokaryotics include inclusion bodies. Very important. Inclusion bodies. And these inclusion bodies are nothing but they store food material in prokaryotic organism. They store food material. And there are several inclusion bodies are present in prokaryotic organisms, like here I am writing, like cyanophycean granule and glycogen and important carboxy and one more polygranule and one more important, that is gas vacuole. So all these five stored their food material, hence all this comes under inclusion bodies in prokaryotes. Even though these inclusion bodies are also amembranous cell organelles. But amembranous cell organelles in eukaryotic means ribosomes, centrioles, and nucleolus. So this is about amembranous or non-membrane bound cell organelle. Now we move on. Very important. That is, what is the role of this ribosome? Role of ribosome play important role in protein synthesis. Hence, they are known as Hence, they are known as protein factory of the cells. Hence, they are known as protein factory of the cell because they helps in protein synthesis by undergoes transcription and translation process from genetic material, that is DNA. Here, how means this DNA undergoes transcription process and then it forms mRNA. So with this mRNA, by using ribosome only, smaller subunit and larger subunit, then it undergoes translation process in presence of ribosomes. So then only it forms the protein. So without ribosome, there is no formation of protein. Hence, ribosome is a factory of protein. This is all about the role of main important role of ribo here. And this ribosome also known as organelle within organelle because the ribosome found inside the mitochondria and chloroplast. Hence, they are known as organelles within organelles. Why? Because this mitochondria and chloroplast itself, they are cell organelles. These two itself, they are cell organelles. And in these two cell organelles, even the ribosomes also present inside this cell organelle. Hence, they are known as organelles within organelle. Yeah. Next, ribosomes. Ribosome is a conjugated protein. It is a conjugated protein, or simply we can use as it is a ribo. Yes, because they made up of with two important structures. Because they made up of with two important structure in which here ribonucleo means here this ribonucleo word this refers to the nucleic acid and protein. Here means nucleic acid and proteinase structures. Nucleic acid proteinase structures together form ribonucleoproteinase structure, that is nothing but ribosome. Here the nucleic acid is always rRNA, ribosomal RNA, and here protein involved is RNA. Now we will move on subunits of ribosome. Generally, in ribosome there are two subunits, Either it is prokaryotic or eukaryotic. They are smaller subunits, and larger subunits. So a complete ribosome which is made up of with two subunits, one is smaller and another one is larger, like this. For example, if you consider this is a smaller subunit and here this is the larger subunit. Larger subunits, this one smaller subunits. And here the larger subunits generally which is bind with very important tRNA. This larger subunits binds with tRNA. Here tRNA means transfer RNA. Generally we have three types of RNA. One is rRNA, second one is mRNA, third one is tRNA. And here tRNA generally bind with larger subunit. And what about smaller subunit? The smaller subunit generally which is binds with mRNA, messenger RNA. Very important, messenger RNA. And one more important thing is here in which magnesium ion very important. And these both unit and smaller subunit and this larger subunit, both units are united, are associated, or their association or dissociation of these both units is depends on the concentration of magnesium ion. Very important. And association and dissociation of these two smaller or larger subunits, it is completely depends upon the concentration of magnesium ion. So here magnesium ion play important role in association and dissociation of these subunits. If generally there is a requirement of a critical concentration of association of this two subunit is 0.01 mole. Sorry, yeah. So here 0.01 mole is concentration is required to bind these two subunits together. If this concentration will decrease, then they get separated from each other. So likewise, the association of subunits and dissociation of subunits is completely depends upon the concentration of magnesium ion. If 0.01 concentration is there means there is an association between two subunits. If there is less than this critical concentration, that is 0.01, then the two subunits are get separated from each other. So finally, each and here both subunits are their association or their dissociation is completely depends upon this magnesium ion, and this magnesium ion play important role. Very important. So likewise, so we have in ribosome two subunits like smaller subunits and larger subunits. Now we will discuss so how and generally these ribosomes are present different in different organisms. We will discuss now. Yeah, here types of ribosomes in prokaryotic and eukaryotic organisms. Types of ribosomes in prokaryotic, eukaryotic organisms. Types of ribosome in prokaryotic and eukaryotic organisms. In prokaryotic, generally there are 70s type of ribosome, and in eukaryotic 80s type of ribosome. And these 70s type of ribosome in prokaryotes are made up of two units. One is smaller subunit, another one is larger subunit, smaller and larger. And this smaller subunit in 70s, that is 30s, here larger is 50s. And here coming to eukaryotic, and it is also made up of with two subunits. One is smaller, another one is larger. And here smaller subunit is 40s in 80s type, and here 60s is the larger subunit. Now coming to smaller subunit in prokaryote, that is 30s, and these has two components, which is made up of with two components. Like here, in which we have 16s rRNA, 16s ribosomal RNA, and one more compound, and these 30s which is made up of with 21 protein. Very important, 21 protein, R protein, 21 R protein. Now coming to larger subunits, and this larger subunit has two components, and here rRNA and R protein. Here the larger subunit has two types of RNA compound. They are 5s rRNA and 23s rRNA. 5s ribosomal RNA and 23s ribosomal RNA. And protein, is 34. There are 34 proteins. Together form 50th type of RNA. So total in 70th type of ribosome, here I am writing, in 70 type of ribosome, here we have 16S ribosomal RNA and 5S ribosomal RNA and 23S ribosomal RNA. Like that we have 3 different types of RNA in 70S type. And the protein is 21 proteins plus 34 protein equal to 55 proteins are present in prokaryotic organism. Okay, yeah. Now coming to eukaryotic organism, and here the smaller subunit is called 40, and this 40S has two components. One is ribosomal RNA. Yeah. So this 40S type of smaller subunit in eukaryote are made up of it. Here they are made up of it 18S rRNA, 18S ribosomal RNA plus there are 33 proteins or protein. 33 R protein. And coming to larger subunit, and this 60S larger subunit has three types of rRNA compound. Here it has three types of ribosomal RNA components. They are like 5S rRNA and 5.8S rRNA, and very important 28S rRNA. So 5S, 5.8S and 28. All these three types of rRNA compounds are present in 60S type of ribosome in eukaryotes. And there are 47 R proteins are present here, in which plus 47 R proteins are present. R protein. So in overview, this 80S type of ribosome in eukaryotes contain different types of rRNA like 18S rRNA in smaller subunit and 5S, 5.S rRNA and 28S rRNA. So these three in larger subunits, and 33 R protein in smaller subunit plus 47 smaller subunit. Sorry, RR protein in larger subunits. So 33 plus 47 means there are 80 R proteins are present in eukaryotic organism, and in prokaryotic organism 55 R proteins are present. So this is all about the types of rRNA, types of ribosome present in prokaryotic and eukaryotic, and their distribution of smaller subunit with respect to their rRNA and their protein component. And here the important point, here S is a S stand for Swedberg unit. They're very important. Yes. So S stand for Swedberg unit, and this Swedberg unit which represent sedimentation coefficient. And sedimentation coefficient means that generally it is indirectly measured of indirectly measures of density and size of the ribosome. Indirectly measure of density and size of the ribosome. And the value for sedimentation coefficient is 10 to the power of minus 13. So this is all about the Swedberg unit, and that stands for S. So with this we have complete about ribosome, and there are two important terms here we need to discuss. So they are polysome and ribozyme. So what these two are? Yeah, here two important concepts we need to discuss. One is poly, are also called poly, and one more concept here we need to discuss, that is ribo, and this is also called enzymatic RNA. Yeah, we'll discuss these two. What all these two represent what? Generally, polysomes are polyribosomes, which is found in prokaryote. Found in prokaryotes. Is found in prokaryote. Here, how these polysomes will form means several ribosomes in which several ribosomes may attach to a single strand of mRNA, messenger RNA, to synthesize, very important, to synthesize multi copies of the same protein, to synthesize multiple copy of the same protein simultaneously by two process. You know that by translation process, sorry, trans process. How means, for example, here by transcription there is a formation of mRNA. If mRNA will form like this means 5 prime to 3 prime, and there are multiple ribosome with attached to this single strand of here, single strand of mRNA like this. Larger subunits, smaller, larger, smaller and larger, like this. So there are several ribosomes, both parts, smaller subunit and larger subunits they attach to this single strand of mRNA to synthesize multiple copies of the same simultaneous protein. Because of these mRNA which undergoes translation to synthesize their respective proteins. And this group of ribosome which attach to the single strand of mRNA, this condition is called poly or polyribosome. Here in word only it represents poly means more ribosomes. So more ribosomes which attach to the single strand of mRNA, and this is the special character which is found only in prokaryotic organism, that is bacteria. Right. Now we'll move on another important concept, that is ribosome or enzymatic RNA. Generally it is a, what is ribosome means, ribosomal RNA means rRNA act as enzyme. Generally rRNA involved in various formation process, like generally they are involved in synthesis of various product, but here this rRNA which act as enzyme. If rRNA act as enzyme, such rRNA is called ribosome or enzymatic RNA. Because why rRNA act as enzyme means, all rRNA not act as an enzyme. Only several rRNA, ribosomal RNA act as an enzyme. In that, most important rRNA is 23S rRNA. Very important. 23S ribosomal RNA act as enzyme. So here 23S rRNA act as which enzyme means peptide transferase enzyme. Peptidyl transferase enzyme, and this enzyme which catalyze peptide bond formation, very important. So this enzyme helps in formation of peptide bond, and these ribosome or enzymatic RNA are non-protein enzyme. Generally enzymes are protein in structures, but except this ribosome is non-enzyme. It is a non-enzyme, non-protein enzyme. Remember that mostly enzymes are protein in nature, but this ribosome is non-protein enzymes. So this is all about the ribosome or enzymatic RNA. So with this we have completed each and every concept regarding related to the ribosome. So that's it for today class, and if you like the video, please hit the like icon and share these videos to your friends, and don't forget to subscribe my channel. So thank you. And here I have provided 3 MCQs from this topic. Yeah, here first MCQs. Yeah, here. So consider the following statement regarding the structure and composition of ribosome. Here asking about structure, sorry, here, yeah, here, structure and composition of ribosome. So read these 4 multi-statement questions. 1, 2, 3, 4 statement I have given, and each of the statement above are correct. Here asking about correct statement. Identify the correct statement. Right. One more MCQ. Sorry. Yeah. So here second MCQs. Yeah. So which of the following statement correctly describe here function? Here asking about function and interaction of ribosomes. These are the keywords in questions. And so here there are 4 options are given. Read this 4 statement, and one more last MCQs. Yeah. Consider the statement regarding the assembly and characteristic of ribosome. Identify the correct statement. Read this 4 statement. Okay. So that's it. So this is all about the ribosome MCQs. So thank you. Have a nice day.